Introduction to particular histology. Nervous system. Nerve, spinal ganglion, spinal cord. Main questions of the lecture. Particular histology is a part of histology studying the microscopic structure of organs, microscopic anatomy. Organs like kidneys, heart, liver, stomach, brain, bladder, ureter, and so on. Organ always consists of all four types of tissues we have studied just before in general histology. By their general organization, plan of structure, all organs can be divided into two main types, parenchymal and line or tubular organs. Parenchymal organs consist of stroma and parenchyma. Stroma consists of connective tissue capsule surrounding organ from outside and layers of loose connective tissue going to the depth of organ. Stroma carries out a supporting, trophic, and protective role. Parenchyma, a highly specialized part of the organ, performing its main specific functions. This is a kidney, an example of parenchymal organ. Another example of parenchymal organ is liver. It also consists of stroma and parenchyma. Stroma including capsule around the organ and layers of loose connective tissue which divide an organ for lobules. Line, tubular organs consist of three membranes, inner, middle, and outer. And membranes consist of layers. So the tissues in line organs organized in layers, and layers form three membranes. For example, in blood vessels, you see here the schematic drawing of artery, half of it, and vein. Another example of tubular organ is stomach and intestine. It also consists of three membranes, the inner tunica mucosa, middle muscle membrane, and outer serous membrane. Every membrane consists of layers. Structural and functional unit of organ. The least part of organ possesses its main functions. For example, muscle fiber, its functional and structural unit of this skeletal muscle. For example, osteon or Gaversov system is structural and functional unit of long bone. A hepatic lobule in the liver. The liver consists of about 500,000 of similar lobules, hepatic lobules. structural and functional units of the liver. The first system of organs you will study in the particular histology part is nervous system. Nervous system may be divided into central part, including brain and spinal cord, and peripheral part, includes nerves, nervous ganglia, nervous plexus, and nervous endings or terminals. Nervous system can be also divided into autonomic and somatic. Autonomic innervates the inner organs and blood vessels, and somatic innervates the skeletal muscles. By their structure, all the organs of nervous system are parenchymal and are composed of stroma and parenchyma. Stroma is composed of connective tissue, include capsule and connective tissue layers, and carries out supporting, protective, and trophic functions. Parenchyma is composed of nervous tissue and carries out the main specific functions of the organs, accepts, analyzes, and transforms information. The first organ is peripheral nerve. It's parenchymal organ. The nerve fibers belong to parenchyma, to the nervous tissue, and connective tissue layer belong to stroma. Those connective tissue layers included Epineurium, the thin, gentle layer of loose connective tissue around every nerve fiber. The connective tissue layer around bundle of nerve fibers called Peripheral, and thick, dense connective tissue layer around the whole nerve called Epineurium. The same similar names like in tendon, endotenon, peritoneum, and epitenon, or in skeletal muscle, endomysium, perimysium, and epimysium. So it's easy to remember. This is a peripheral nerve transverse section stained by hematoxylin and eosin. You see individual nerve fibers. They're myelinated in this case. They look like light rings because myelin chains doesn't stain by eosin. And point inside is axial cylinder, process of neuron axonodendrite, and around endoneurium and bundle of nerve fibers surrounded by perineurium and whole nerve covered by epineurium, dense connective tissue. Is the same transverse section of nerve stained by osmium, which specifically stain lipids or myelin chains. At high magnification you can see the individual myelinated nerve fibers like black rings with axial cylinder in the center, not stained, slight. You can see also unmyelinated nerve fibers, like much smaller and lighter rings or light spots. Endoneurium, perineurium, and epineurium doesn't stained by osmium. On this scheme you can see the peripheral nerve or spinal mixed nerve. Nerve fibers here included dendrites of sensory neurons. They are myelinated and compose receptors at the periphery. And axons of motor neurons, also myelinated, compose the neuromuscular junction in the periphery. So this is a reflex arch on the picture. This is a spinal cord, the sensory ganglion. It's spinal ganglia. It contains sensory pseudo-unipolar neurons. The dendrites go into the periphery, receptors, and axon forming the posterior root and connected with intermediate neuron of posterior horn of spinal cord, or directly with motor neurons. Spinal ganglion is also parenchymal organ, so it consists of stroma and parenchyma. Stroma represented by capsule, dense connective tissue surrounding ganglion, and a gentle loose connective tissue layer which going inside ganglion, and they surrounded every sensory neuron. Parenchyma consists of nervous tissue. Nervous tissue consists of neurons and neuroglia. Neurons, sensory, pseudo-unipolar neurons. They have large, round pericardions, neuronal bodies, located by groups, mostly the periphery of the ganglion, and nerve fibers, mostly myelinated. Dendrites of these neurons go into the periphery, and axons compose the posterior root and going inside spinal cord. So this is posterior root and this is anterior root. And anterior and posterior root fuse together and compose the mixed peripheral nerve. Autonomic ganglia, including the group of neurons. They may be located outside the inner organs, extramural autonomic ganglia, and inside organs, intramural autonomic ganglia. So they belong to autonomic nerve system. Central nervous system consists of brain and spinal cord. Spinal cord consists of 4 parts: cervical, some thicker part, thoracic, the longest, lumbar, also some thicker, sacral, and coccygeal, very short segment. And at the picture, the spinal ganglia are visible beside of spinal cord. Spinal cord consists of 31 segments. Every segment included 2 pairs of roots, pair of posterior roots and pair of anterior roots, and pair of spinal ganglia. Spinal cord in transverse section, its parenchymal organ, consists of stroma and parenchyma. Stroma represented by capsule around, which consists of meninges, dura mater, arachnoid, and pia mater, and a loose connective tissue septa with blood vessels going inside. Spinal cord consists of 2 symmetric parts, halves. In the center of spinal cord, situated the gray looks like matter, butterfly or letter H. It consists of thicker anterior horns or ventral horns and thinner posterior or dorsal horns. Gray matter consists of neuronal bodies and glial cells bodies. Neurons in gray matter spinal cord organized in nuclei, the groups of neurons similar in structure and functions. For example, in anterior horns you can see the motor nuclei, including the groups of motor neurons, effector neurons. The axons are forming the anterior roots and fuse the posterior roots and compose the mixed peripheral nerves. All neurons in spinal cord are multipolar. In anterior horns they are effector motor neurons. In posterior and lateral horns are intermediate neurons. Posterior roots composed by myelinated nerve fibers, bundles of axons of sensory neurons of spinal ganglia. In the center, you can see the central canal lining by ependymal cells. A liquor, a cerebral spinal fluid circulate inside. The neurons of spinal cord surrounded by satellite oligodendrocytes. There are also astrocytes, the bodies of astrocytes in gray matter. Their processes going to the periphery and build the glial membranes. White matter around consists of 3 funiculus: anterior, between anterior horns, posterior, between posterior horns, and lateral. White matter consists of nerve fibers, mostly myelinated nerve fibers. You can also see here the gray commissure connecting the gray matter and white commissure connecting the white matter. This is the anterior fissure. These pictures demonstrate the motor neurons of anterior horns of spinal cord, staying for neurofibrils by impregnation and staying for chromatophilic substance, nasal bodies demonstration. Autonomic nervous system. It's part of nervous system, consists of two parts, sympathetic, left side, and parasympathetic division, right side. They both together innervate every organ of the body. Both of them consist of central and peripheral parts. Central part centers of sympathetic nerve system located in lateral horns of spinal cord, in thoracic and upper lumbar part. Their neurons, axons, forming the preganglionic fibers. They terminated on the secondary neurons, which located in autonomic nerve ganglia, in sympathetic nerve ganglia, in celiac ganglion of abdomen cavity, or in pelvis. And these secondary neurons, their axons, going to the inner organs and compose postganglionic nerve fibers. Centers of parasympathetic system, partly located in lateral horns of spinal cord, but only in sacral part, and others located in brain stem, in medulla oblongata and midbrain, and their axons of those neurons compose also preganglionic fibers, and then in nerve ganglia secondary neurons located, and the axons of those neurons compose the postganglionic fibers. They terminated also in organs. So every organ has double sympathetic and parasympathetic innervation. Reflex such as somatic, autonomic, and local. You know, somatic. Afferent part include sensory neurons of spinal ganglia. Afferent part include the effector neurons of anterior horns motor neurons. Autonomic nerve system, both sympathetic and parasympathetic, has the similar afferent part. It's also composed of sensory neurons of spinal ganglia, but efferent part consists of two neurons. First, located in lateral horns of spinal cord or in brain stem, and second, located in nerve ganglia outside or inside the organs. In addition, inside organs there are local reflex arches belong to a metasympathetic nerve system. It includes special neurons inside intramural ganglia, type 1, effector, motor or secretory neurons, and Doggle type 2 are sensory neurons, which start by receptors and finish by synapses on the doggle. They're located inside inner organs, and they provide nerve regulation of the organs, and even after cutting all nerves going to the organ. This picture demonstrates Doggle's type 1 effector neurons with long axons and short branches dendrites, and Doggle type 2 neurons with equal processes. These are sensory neurons. So these two types of neurons can compose simple reflex arches inside inner organs. Early period of human nervous system development. It starts from 19th day of embryogenesis. This is the embryonic disc, the surface and transverse section. You see here neuroectoderm. This nerve plate, neural plate. Then it invaginated and nerve groove appeared. You see this groove, and from 22nd day the edges of nerve groove fuse together and forming the nerve tube, initially in the middle of embryo, then it became longer, and from anterior part the brain developed, and from posterior part the spinal cord developed. There are also the group of cells between neural tube and ectoderm called nerve or neural crest. These cells is the source of neurons and neuroglia of the peripheral nervous system. They compose, for example, nerve ganglia and plexus. The third source of nervous tissue and nervous organs development are placodes. The thicker part of ectoderm. They are the source for sensor epithelial cells of organ of hearing and equilibrium development. This is a human embryo schematic drawing at the end of the first months of development. You see ectoderm around and nerve tube